Hello, welcome to Learning Theatre. In today's video, we're going to talk about glycolysis. And when we talk of glycolysis, it's a central metabolic pathway that breaks down glucose into pyruvate and producing energy in the form of ATP and reducing power in the form of NADH. And this glucose that we are going to break down into pyruvate is a monosaccharide, a 6-carbon molecule. And this we get it from our diet. So we are going to oxidize the glucose into two pyruvate, that is two 3-carbon molecules, because each of the pyruvate is a 3-carbon molecule. So you're going to get two 3-carbon molecules. But how do we get glucose into the cell? I will take you through that. And glucose is a water-soluble solute which cannot move through the cell membrane by diffusion. It has to come through by some type of transporters. And then this is our glucose here outside the cell. So for it to be able to get into the cell, it needs some transporters that will transport it into the cell. And then this is our transporter. So let me name this one glucose. So this is our glucose molecule. So this transporter here is what is going to transport glucose from outside the cell into the cell. And then with this transporter, we have so many types of them. We have GLAT1 transporter. Let me write that down. GLAT1 transporter or receptor. Let me use receptor. GLAT2 receptor. GLAT3 receptor. GLAT4 receptor. So these are the types of receptor or the type of transporters that we have that can transport glucose from outside the cell into the cell. And GLAT1 transporter can be found in the red blood cell, fetus, and then in blood-brain barrier. Whereas GLAT2 receptors can also be found in the kidney, liver, and then the pancreas. We can also find GLAT3 receptors in the placenta, neurons, and then the kidneys. And then the GLAT4 receptors can be found in the muscles and then the fat or the adipose. Yes, so apart from GLAT4 receptor, apart from the GLAT4 receptor, the remaining type of the receptors or the transporters are um, are insulin independent. They are insulin independent, and then GLAT4, meaning that when we say they are insulin independent, it means that they don't depend on the concentration of insulin. They don't depend on the concentration of insulin to increase in their number or their type of receptors. But GLAT4 is insulin dependent. This means that it needs insulin, or insulin help it to increase in the number of GLAT4 transporters. So it requires insulin in order to increase in the number of its transporters. Yes, so it is the only receptor or GLAT receptor that need insulin to increase in its number or its concentration. So this glucose here is brought into the cell by one of these transporters depending on the type of organ. So these transporters, they are bidirectional. They take glucose in and can also take glucose out. So they are bidirectional. And so it means that when glucose comes into the cell, it is capable of moving out again. So in order to prevent this glucose from moving out of the cell, we add a phosphate group at the carbon 6 of glucose. when glucose is now brought into the cell. In order to prevent it from moving back, we add a phosphate group at the carbon 6 of glucose. So this is the phosphate group that has been added. So we have carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, carbon 6, where we added the phosphate group. And then adding the phosphate group to glucose, we form a new molecule known as glucose 6 phosphate. Glucose 6 phosphate. Let me write that down. So we have glucose 6 phosphate. Glucose 6 phosphate. And this is done by an enzyme known as hexokinase 2 or glucokinase. Hexokinase 2 or glucokinase. Let me write that down. So hexokinase 2 or glucokinase. And then it is catalyzed by hexokinase 2 or glucokinase, depending on the type of organ. So hexokinase can be found in different types of tissues, like the muscles and then some other tissues. And then glucokinase is only catalyzed, is only needed when it is in the liver, when it has to do with the liver. So apart from the liver, all the other tissues, it is catalyzed by hexokinase enzyme. But in the liver, it is catalyzed by glucokinase. And then liver is the only one having glucokinase that is catalyzing this reaction. So one of these enzymes will convert glucose to glucose 6 phosphate. But where does the phosphate comes from? This phosphate, we get it from taking ATP and then converting it to ADP. So we have adenosine triphosphate being converted to adenosine diphosphate. So we had tri and then now di. So meaning we've lost one. We are having three phosphate now turned to two phosphate. So we've lost one. So adenosine diphosphate triphosphate converted to adenosine diphosphate, meaning we lost one phosphate group. And this phosphate is used to add to this glucose to form glucose 6 phosphate. So we get the glucose from the conversion of ATP to ADP. An enzyme known as phosphohexoisomerase comes in and then convert glucose 6 phosphate to fructose 6 phosphate. Let me write that down. So fructose 6 phosphates. And then the name of the enzyme is phosphohexoisomerase. So phosphohexoisomerase. So this enzyme will come in and then convert glucose 6 phosphate to fructose 6 phosphate. And glucose 6 phosphate and fructose 6 phosphate, they all have the same number of carbons and then hydrogens, but glucose 6 phosphate is usually an aldehyde, whereas fructose 6 phosphate is usually a ketone. And then this enzyme, phosphohexoisomerase, convert it from the aldehyde to a ketone. An enzyme known as phosphofructose kinase 1 will come and then add a phosphate to carbon 1. Would come and then add a phosphate group to the carbon 1. You see, we already have a phosphate group at the carbon 6. So this enzyme, phosphofructose kinase 1, will come and then add a phosphate group to the carbon 1 to form a new molecule known as fructose 1,6 bisphosphate. So let me write the name of the enzyme down. Phosphofructose kinase 1. This enzyme will add a phosphate group to the carbon 1 so that we now have two phosphate groups, one on carbon 1 and then one on carbon 6, which will cause the molecule to change, and then we now get a new molecule which is known as fructose 1,6 bisphosphate. It is known as bisphosphate means that there are carbon spaces between the phosphate group. There are carbons between the phosphate group, so that's why we call it bisphosphate. When is uh, when it's a biphosphate, it is known as and biphosphate means that they are right next to each other. Biphosphate means that they are right next to each other. So if like one phosphate is on carbon 1, the second phosphate will be on carbon 2, and then that one is a biphosphate. But when there are different many or spaces between the phosphate group, we call that a bisphosphate. Now, fructose 1,6 bisphosphate will split into two 3 carbon fragments. It will split into two 3 carbon fragments. So you have one here, and then the other one two here. So we have two 3 carbon fragments. Yes, and then each fragment is having a phosphate group attached to it. So this is the phosphate group here, and then the other phosphate group is also attached to this one too. And then the name of this 3 carbon group is known as the name of this 3 carbon molecule here is known as dihydroxyacetone phosphate. Dihydroxyacetone phosphate. And the other 3 carbon molecules is also known as glyceraldehyde 3 phosphate. Glyceraldehyde 3 phosphate. And this is an irreversible reaction. Irreversible reaction. Yes. And this dihydroxyacetone phosphate is not utilized in glycolysis pathway. Therefore, it is this dihydroxyacetone phosphate needs to be converted to glyceraldehyde 3 phosphate in order to go through the glycolysis pathway. So meaning that from here downwards, we're going to go through the glycolysis pathway twice from here downwards, because we're now going to have two glyceraldehyde 3 phosphate, and each one of them is going to go through the glycolysis pathway, giving us two of them. So anything we are going to do next is going to be 2, 2, 2, 2. The name of the enzyme that involved in the splitting of fructose 1,6 bisphosphate into dihydroxyacetone phosphate and glyceraldehyde 3 phosphate is known as idolase. So the name of the enzyme is called idolase enzyme. And the name of the enzyme that convert dihydroxyacetone phosphate to glyceride phosphate is known as triose phosphate isomerase. So let me write that one down. Triose phosphate isomerase. Isomerase. So this enzyme converts dihydroxyacetone phosphate to glyceride phosphate. The next thing is that this glyceride phosphate is converted to 1,3-bisphosphoglycerate. 1,3-bisphosphoglycerate. Let me write it down. 1,3-bisphosphoglycerate. 1,3-bisphosphoglycerate. And it has a phosphate on carbon 1 and then the carbon 3. So you can see that, and here we are having only one phosphate group on the carbon 1, but coming here we added another phosphate. Now we have one phosphate on the carbon 1 and then the other phosphate on the carbon 3. And mind you, I said bisphosphate means that there are carbon spaces between the phosphate group. So this one is also a bisphosphate. And this is done by an enzyme known as glyceraldehyde phosphate dehydrogenase. Glyceraldehyde phosphate dehydrogenase. So glyceraldehyde 3-phosphate dehydrogenase. So this enzyme will convert glyceride 3-phosphate to 1,3-bisphosphoglycerate. And in this process, NAD plus react with glyceride 3-phosphate in the presence of this enzyme, glyceride 3-phosphate dehydrogenase, and it rip off the hydride and then convert it to NADH. So in this reaction, there is conversion of NAD plus to NADH. Let me write that one too down. So we have NAD plus to NADH. And then remember, I said from here onwards, we're going to run the pathway twice. So meaning that we're going to have two NAD plus being converted to NADH. And the hydride I used here means that a proton plus two electrons, and this is how it is and proton, and then you add two electrons. So this is what we call a hydride. And then and now with 1,3-bisphosphoglycerate, we now convert ADP to ATP. But because the reaction is run twice, because this reaction is run twice, we are producing two ATPs, meaning that two ADP is being converted to two ATPs. So let me do that one too. So we have two ADP converted to two ATP. Yeah. And this is done by the enzyme known as phosphoglycerate kinase. Phosphoglycerate kinase. Let me write that down. Phosphoglycerate kinase. This kinase is something that phosphorylate a substrate. So in this case, it is phosphorylating ADP and is getting its phosphate from the 1,3 base phosphate. 1,3 base phosphate. Yes. So giving it to ADP, it make ATP. It is getting its phosphate group from the 1,3 base phosphoglycerate, and we now form a new molecule which is known as 3-phosphoglycerate. 3-phosphoglycerate. Let me write that down. So you have 3-phosphoglycerate. 3-phosphoglycerate, and the phosphate group is moved from the third carbon. Look at here, it was on the third carbon. So this phosphate group here, we now move it from the third carbon here to the second carbon, of the next molecule. And this is done by the enzyme known as phosphoglycerate mutase. Phosphoglycerate mutase. So let me write that down. So phosphoglycerate mutase. This will move the phosphate group from the third carbon to the second carbon, and then this will form a new molecule known as 2-phosphoglycerate. 2-phosphoglycerate. So 2-phosphoglycerate. You see, when the phosphate was on carbon 3, we called it 3-phosphoglycerate. And then the phosphate now comes to carbon 2. We now call it 2-phosphoglycerate. And an enzyme which is known as enolase will also come and then convert 2-phosphoglycerate to a new molecule known as phosphoenol pyruvate. Phosphoenol pyruvate. So this one here is called phosphoenol pyruvate. This is catalyzed by the enzyme enolase. So the name of the enzyme that will come and then convert 2-phosphoglycerate to phosphoenol pyruvate is known as enolase. So enolase enzyme. And the enzyme known as pyruvate kinase will come and then take 2-ADP and then react with the phosphoenol pyruvate. Oh, sorry, my camera wasn't showing. So, like I was saying, this 2-phosphoglycerate is being converted to a new molecule known as phosphoenol pyruvate. And this is done by the enzyme enolase. Yes, so then phosphoenol pyruvate, an enzyme known as pyruvate kinase, will take 2-ADP and then react with this phosphoenol pyruvate and then make 2-ATPs. So let me do that. It will take 2-ADPs. So 2-ADP and then convert it to 2-ATP in the presence of the enzyme pyruvate, the enzyme known as pyruvate kinase. So this enzyme is the one that is going to take two ADPs and then react it with phosphoenolpyruvate and then convert the 2 ADP to 2 ATPs. They are 2 because it is 2 ADP converting to 2 ATPs because this reaction is being run twice. So let me write the name of this enzyme down. The enzyme is known as pyruvate kinase. Pyruvate kinase. So I want you to observe something. From the start, we had most of our arrows in color blue, and we only had 3 arrows which are in color violet. And then this reaction or this arrow signifies an irreversible reaction, meaning that you can't go back. So it's an irreversible reaction. And the other one was from glucose to glucose 6 phosphate, and the other one was also from fructose to the other one was from fructose 6 phosphate to fructose 1,6 bisphosphate, and then the last one is from phosphoenolpyruvate to pyruvate. So these reactions are the irreversible reactions in glycolysis. And so this enzyme pyruvate kinase here is a highly regulated enzyme, and it is also this step, and this step is also an irreversible reaction. Like I said, you can't go back through this enzyme. So it's an irreversible reaction. And then this enzyme takes the phosphate from the phosphoenolpyruvate and then convert it to pyruvate by transferring the phosphate from phosphoenolpyruvate onto the ADP to make 2 ATPs. So remember, because we had 2 glyceride 3 phosphate, we now made 2,1,3 bisphosphate, and we also made 2,3-phosphoglycerate, and then we had 2 um 2 phosphoglycerate, and we also have 2 phosphoenolpyruvate and then 2 um pyruvate molecule. So let me write that down to 2 pyruvate. This one is a pyruvate molecule. So pyruvate. The last one molecule is known as pyruvate. Yes. This pyruvate molecule is a 3-carbon molecule, and it can divert to 2 different pathways. And then so what is the actual fate of this pyruvate molecule? The actual fate of this pyruvate molecule is that it can get converted to 2 molecules. The one is during anaerobic condition and or no oxygen, anaerobic condition or no oxygen, and the other one is during aerobic condition or when there is oxygen. And then NADH are always to load their hydride on certain molecules so that it can be taken to the electron transport chain to produce ATP. But during anaerobic condition, this NADH, they don't load their hydride to those molecules, but they load them on pyruvate and then get converted to NAD plus. So when they load their hydride to pyruvate, they will convert to NAD plus. So we're now going to have 2 NAD plus, and they will get oxidized. But this pyruvate get reduced. So when NAD plus is oxidized, the pyruvate will get reduced and get converted to a molecule known as lactic acid. So this is the lactic acid here. It's also a three-carbon molecule. So lactic acid. Yes, lactic acid. And the name of the enzyme that is involved in converting pyruvate to lactic acid is known as lactic dehydrogenase. So lactic dehydrogenase. Dehydrogenase. This enzyme will convert pyruvate to lactic acid, and this lactic acid can go into the liver and get converted into glucose or be used to make ATP. So lactic acid can be can cause the pH to increase, and it also makes the blood more acidic. Yes, so that's the lactic acid. And pyruvate can also be converted to another molecule known as acetyl CoA. Acetyl CoA. Acetyl CoA. We're going to talk about this one in our um, next video. That is the transition step. Yes, so we'll talk more about this acetyl CoA in there. And so we learned that pyruvate can be converted to lactic acid or acetyl CoA, and acetyl CoA is also known as the transition step. So we talk more about it in the transition step video. So in summary we talked that glycolysis occurs in the cytoplasm, and glucose is a certain substrate, and then we said glucose is brought into the cytoplasm and then is phosphorylated to prevent it from moving back. And we said our end product that we make from glycolysis is 2 pyruvate, and we also make 4 ATPs, 4 ATPs, where we use 2 ATPs, leaving a net ATPs of 2, and we also make 2 NAD. And also during anaerobic process we generate lactic acid. Thank you for watching. Please like the video, subscribe, and then comment, and also share this video to your friends.